Hello, I'm Margaret Ackerman from Dartmouth College, and it's my pleasure today to speak to you about polyfunctional HIV-specific antibody responses and their associations with spontaneous viral control. So we undertook this study with the notion that elite controllers, subjects who are HIV infected but spontaneously able to suppress virus to below the level of detection, might be a model of a functional HIV cure and might possess humoral immune responses that are desirable to induce via vaccination. And our goal was to understand the characteristics of these antibody responses and how they relate to effective viral suppression, specifically in terms of their ability to interact with innate immune effector cells. So one of the ways that antibodies mediate antiviral activities is by binding directly to a virus and neutralizing it, but in conjunction with that activity, or even in the absence of direct viral neutralization, antibodies recruit a host of innate immune effector cells drawn here on the screen, including monocytes, phagocytes, natural killer cells, eosinophils, neutrophils, dendritic cells, all which possess a wide range of different mechanisms for handling and destroying antibody-coated viral particles or virus-infected cells. And there's been a lot of great work over the past 20 years suggesting that spontaneous viral control might be associated with unique types of antibodies that are better able to recruit these innate immune effector mechanisms of viral clearance. So some of this evidence came from early studies by Linda Baum, who found that ADCC-inducing antibodies were more prevalent in subjects who did not progress rapidly to AIDS and had CD4 counts, higher CD4 counts over longer periods of time. Other evidence has been genetic evidence. So an FC receptor genotype, FC gamma R2A alleles. These are some of the innate immune receptors expressed on effector cells that interact with and recognize antibodies. Different alleles of these receptors have been associated with different rates of progression and variable CD4 counts. Lastly, in the case of responses to vaccination, non-neutralizing antibodies that can induce antibody-dependent cellular cytotoxicity and natural killer cell-mediated antiviral function were correlated with post-infection viral control. So there's been 20 years of studies suggesting the potential involvement of functionally potent antibodies playing a role in long-term viral suppression. One of the most potent ways that antibody effector function is modulated is by IgG subclass. So humans have 4 IgG subclasses, and they exhibit dramatically different potencies in their cytotoxic properties. So this plot shows the percent of NK cell-mediated killing of an antibody-coated target cell, and you'll see that IgG3 and IgG1 antibodies mediate potent killing of an antibody opsonized cell, whereas IgG4s and IgG2s are multiple orders of magnitude less capable of inducing this potent antiviral activity. Interestingly, these subclass differences also impact neutralization potency. So in a study by Scharf, polyclonal antibodies were fractionated into those comprised of IgG3s and compared to a polyclonal pool of IgG1 antibodies. And when these antibodies were tested as monovalent FABs, they exhibited equivalent neutralization potency. That's what's shown here. But when the whole IgGs were evaluated, the IgG3s exhibited approximately a half or an order of magnitude better potency at inducing neutralization. So this implies that antigen recognition was consistent between these two pools of polyclonal antibodies, and yet neutralization was more potent in the IgG3 pools. And this might have something to do with some of the unique structural aspects of the different subclasses. So here I've shown a schematic of the IgG3 antibody type, which has been described to have an increased range of motion for its FAB arms relative to each other and relative to the FC domain. as well as an extended hinge region that might also impart different reach and range characteristics. And I'll be talking about IgG4 responses later, and IgG4s are uniquely characterized by this process called Fab-arm exchange, where actually the two halves of the antibody can dissociate from each other and spontaneously reform. And what this ends up resulting in is a monovalent recognition of antigen. Instead of being a bivalent antibody, the two arms have spontaneously dissociated and reformed with other IgG4 specificities. So we wanted to follow up on these early studies, and we wanted to ask, what are the antibody effector profiles that elite controllers have? Taking a more in-depth look across many different antiviral functions than had been previously characterized. And then beneath that, we also wanted to determine what were the specific characteristics of the antibodies that possessed these antiviral activities. What are the features of functionally potent antibody responses that we might wish a therapeutic monoclonal or a potent vaccine-elicited response to recapitulate? And so the way we undertook this was to profile the biophysical characteristics of the polyclonal antibodies present in different HIV-infected populations, and to marry that with functional assessments using a wide variety of effector cells, and then to look for associations within these two data sets. So we compiled a cohort of 200 HIV-infected subjects, which were balanced for age, gender, and the presence of protective HLA alleles. We evaluated 7 different antibody effector functions, including those mediated by natural killer cells, phagocytic cells, and the induction of the complement cascade, or CDC activity. And the biophysical characterization we evaluated in this study included an assessment of antibody titer, or the level or magnitude of the antibody response, as well as a qualitative assessment of which subclass of antibodies were present in each sample. So in terms of the functional profiles that were observed in the antibodies from these elite controllers, we were surprised that we did not see enhancement in any functional activity in the elite controllers as compared to any of the other subject groups, which included viremic controllers. They're noted as VC, chronic treated subjects, and untreated subjects, CTs and CUs. If anything, the elite controllers tended to often exhibit a somewhat lower effector function activity. So this was surprising, but perhaps due to or related to the fact that this controller cohort has not progressed for many, many years, and some of the antibody responses are waning. What we did notice was different about the elite controllers, though, was that their antibodies were able to elicit the antiviral activities of multiple different effector cell types. So what's shown here is the correlations between pairs of effector functions. And the degree of correlation is reported in this pie chart. So highly correlated functional responses are shaded in black, and pairs of responses that were not correlated at all are in white. And you can see that there was a greater extent of functional coordination in the antibodies present in the elite controllers. So this implied that they may have had fewer antibodies present, but those antibodies were able to recruit NK cells, complement all the different mechanisms of antiviral activity quite well, as opposed to antibodies present in some of the other subject groups, which had more monofunctional profiles. The polyclonal pools of antibodies could elicit the activity of NK cells or monocytes, but not necessarily both. So looking at titer, we did observe a somewhat lower level or magnitude of antibody responses present in the elite controllers in terms of the total magnitude of IgG responses. So it was interesting that we observed the polyclonal profile despite a lower overall level of antibody response. So to dig into the characteristics of these antibodies more deeply, we looked at the fraction of subjects that were positive for each of the different antibody IgG subclasses. And what we observed here was that across all subject groups, IgG1 positivity was common, and there was a striking absence of IgG4 antibodies among elite controllers, a low level of response among viremic controllers, and up to approximately half of the chronically infected subjects were positive for IgG4 antibodies. So we observed some differences in positivity among the subclasses. To look at the magnitudes of these responses, again, we observed IgG1 responses, which make up the bulk of the antibody IgG response, were lower in elite controllers, as were some of the other subclasses. But again, we observed much lower levels of IgG2 and IgG4 responses among the spontaneous viral suppressors. So the qualitative characteristics of the antibodies across these different subject groups were divergent. We next looked at patterns within the subclasses. So I noted before that IgG3 and IgG1 antibodies are potently active. They have high effector function, whereas IgG2s and IgG4s exhibit reduced binding to Fc receptors and reduced effector function. And what we could observe across our cohort was that IgG3 and IgG1 responses were coordinately induced, whereas the two antibody types, IgG2s and IgG4s, with lower activity were also induced in correlation with each other. So we could see evidence of the immune system making a choice between inducing more or less potent antibody types. We next wanted to look into each of these different subclasses and look at their correlations with the functional activities that were measured. So this plot is a heat map that describes the degree of correlation between each antibody measurement and each function in each of the 4 HIV-infected subject groups. And what you'll notice strikingly is that there wasn't a whole lot of correlation between any of these measurements and the antibody activities in several of the subject groups. And that's in stark contrast to the levels, the relationship between antibody levels and antibody functional activity in elite controllers, where we saw very strong associations between the level of IgG3 responses and many different antibody functional activities. So though the IgG3 antibodies were low in prevalence, and though the overall antibody response was lower in elite controllers, these IgG3s were associated with multiple antibody effector functions. And the antibodies present in many other populations did not seem to predict or correlate with activity well. In fact, some antibody types correlated inversely with the antibody functions. So here we took a ratio of the level of each subclass relative to the overall magnitude of the response in each subject. And what we observed in this analysis was a strong inverse correlation for the level or the relative level of IgG2 and IgG4 antibodies and each of the functional measurements. So this indicated that these two antibody types were associated with reduced functional activity and might have an actively negative impact on the composite activity of a polyclonal serum sample. What we next did was use this subclass data to build models that predicted the degree of polyfunctionality observed in each of the subjects. And when we used these subclass measurements to predict polyfunctionality, we found that IgG1 and IgG3 levels were contributed positively to polyfunctional activity. And again, the levels of IgG2 and IgG4 responses gave a negative contribution to prediction of polyfunctional activity. So more support that these subclass types can actively reduce functional activity. And also an indication that the relative composition of the polyclonal antibody pool may be a more important characteristic in terms of defining antibody function than the magnitude of the antibody response. So in conclusion, for this part of the talk, antibody quality can be more important than quantity. So despite having lower titers, generally across the board of all antibody types, the antibodies present in elite controllers were higher quality, more polyfunctional. And some antibody pools are polyfunctional, capable of recruiting multiple effector types and using different antiviral mechanisms, whereas other polyclonal pools are really only effective at recruiting one type of innate effector and not others. We also observed evidence that the functional profile of antibodies are coordinated at the level of subclass selection. So these results are relevant for monoclonal antibody therapy and for vaccines in that they point to the characteristics of the desirable characteristics of polyfunctionality and IgG1 or IgG3 biased immune responses. And we've also observed important patterns regarding subclass induction in vaccines. So in the RV144 vaccine trial, IgG3s were a correlate of reduced risk. So what's shown in the plot at the left in a study by Yates et al., IgG3 positive subjects exhibited a lower rate of infection than vaccinated subjects who did not produce IgG3 antibodies. What's shown at right in a study by Chung et al. Is the correlation between each of the antibody subclasses. So this first panel is IgG1, 2, 3, and 4 across a set of 6 different antibody activities. And what you see here is that the IgG1 and 3 responses were associated positively with each of these functions, whereas again, IgG2s and 4s, same as we observed in the elite controllers and the chronically infected subjects, were negatively associated with these activities. So what was done after these associations were observed was to look at whether they were mechanistically relevant. So what we did next was to take the polyclonal pools of antibodies from the RV144 vaccine recipients and to deplete those samples of the IgG3 pool. And when that was done, for both phagocytic activity and for NK cell-mediated cell killing, we observed a striking decrease in antibody activity. So despite the fact that the levels of IgG3s were rather low, a great deal of functional activity was lost when this minor component of the plasma pool was depleted. The same thing was done in terms of depleting IgG4s, and what was observed in this case was that functional activity was increased when the IgG4s were removed from the polyclonal pool. So this was direct evidence that more antibody is not always better, and more IgG4s actively reduced the activity of the rest of the antibody pool. And this is pretty important because in looking at many of the vaccine strategies that have been evaluated recently, they include multiple protein boosts. And what's been observed in these studies is that many of the recipients shift their antibodies towards IgG4, away from IgG3s and towards IgG4, which is a type of response that's not very prevalent and not at a high level in natural infection. So what's shown here are the IgG4 levels across a set of infected subjects in comparison to vaccinees from the VAX-004 trial. And you can see a much higher level of IgG4 responses present in vaccinees. And many of the strategies that are being evaluated in preclinical models involve repeated protein boosts. And so to the extent human vaccines have evaluated the effect of repeated boosts, they've observed a striking shift away from 3s and towards IgG4s. That makes some sense if you look at the locus, the IgG locus, and what you'll see here is that it's arranged in order, kind of in order of potency. So IgG3s are the first subclass at this locus, followed by 1s, and the last 2 IgG types are IgG2 and IgG4. And so over time, in an acute response in acutely infected subjects or after early immunizations, IgG3s are prevalent. And following subsequent boosts or elapsed time of infection, this response can shift towards the less functional antibody types. However, it's thought that repeated immunizations and longer time periods will be important for maturing antigen recognition. So for improving the affinity of the interactions between antibodies and virus, and for increasing the level of mutation that those antibodies evolve toward as they gain breadth and potency in terms of viral recognition and viral neutralization. So there might be competing factors at play. As we boost, we might make antibodies that are better at viral recognition, but poorer at actually clearing antibody-coated virus or antibody-coated virally infected cells. So in summary, if we take our findings in the study of elite control and what's known about effective vaccines, we can find evidence from both settings that antibody quality may be more important than antibody quantity. So response characteristics rather than response magnitudes may dictate antibody efficacy in vivo. And it might be really important to find means to stably fix and recall IgG3 responses even when repeated protein boosts are administered in the setting of vaccination, or at least avoid the induction of IgG4s, which we've shown can actively reduce the antiviral activity of a polyclonal immune response. And with that, I'd like to thank the collaborators in this study, Galit Alter, whose group conducted all of the functional analysis, and the Reagan Institute, whose cohort of subjects were evaluated here. We'd like to thank the study participants, as well as the Bill and Melinda Gates Foundation and the NIH for supporting this research. Thank you.